Welcome to the K-State Regen Ag Pod, where we talk all things regenerative agriculture here at Kansas State University, across the state of Kansas, and beyond. I'm Nick Detter, your host, and today I'm joined by Dr. Lydia Zeglin, Kansas State University Professor of Biology and University Outstanding Scholar. Her research focuses on microbial ecology, microbial diversity, and nutrient cycling in streams and soils. She joins us today to discuss that research. Lydia, thank you for joining us.
Great. Yeah, thanks, Nick, for having me. It's fun to be here.
Well, I think just to start, if you could just introduce yourself to our audience a little bit, talk about your background and describing your current role at K-State.
Yes. I've been at K-State for 12 years, almost exactly, and joined the biology department as a microbial ecologist. So that means I study small organisms, micro, that you can't see with your naked eye, and how they interact with their environment, which is ecology. To get here, I've went many places through sort of the journeyman process of being an academic researcher. But I'm from Wisconsin. My parents still have a small livestock farm in west central Wisconsin. Although my dad went back to college and got a teaching degree, so we could move to a bigger, move from the 10 people in the classroom sort of size, which is awesome, but went to a really large high school. And after that, my parents moved back to the farm. College in Madison, Wisconsin, and then studied ecology there, and then biology PhD in New Mexico, also in a biology department, but that was really studying water quality and arid grassland influences of microbes on the soil and water quality and health and those systems. Worked in Oregon for 4 years in a soil science department as a in the microbiology group in the soil science department. I studied nitrifiers and fungi there. 1 year in Alaska, and then came back here to Kansas. So this is actually the longest I've lived anywhere, but really fortunate to have this cool job with a lot of opportunities for research and working with people in different systems, locally and in other places.
So what would you say sparked your interest in microbiology?
Oh, that was a long path too. I actually went to college to study wildlife ecology. I think because I just wanted to be outside as much as I could. I took all the field classes, field identification of whatever. That's what I would gravitate towards. But as I learned more and started thinking about the interactions between all of these plants and animals in the system, I started putting the pieces together to realize that it was the organisms we can't see living in the soil and the water that are really affecting how well the plants and animals can grow. So it's really like the curiosity of digging deeper into those connections, I think, that made me go to grad school in the first place, which I never thought I would do, and study microorganisms specifically.
Yeah, and now that you're in a research position, a professor, and what are some of your broad research topics that you've been looking at and the types of questions that interest you and that you try to study?
Yeah, there's a lot, which again, I said was pretty cool, but we think about the intersection between the tiny organisms and the whole ecosystem. So how do bacteria and fungi and other tiny creatures change the environment around them just by breathing? In doing so, they breathe in certain chemicals, breathe out or excrete certain chemicals, and that affects nutrient availability for plants to grow, for example, and also removes excess nutrients from water. That nutrients can become pollution and too high of a level. So in the water system, the microbes or microbiomes are helping remediate nutrient pollution, and we study how specific types do that through their lifestyles. And then in the soil, we study how specific types of microorganisms release nutrients from organic matter to help feed plants. But the big questions lie in which type of organism is performing which important function for the larger scale system.
Well, you mentioned that larger scale system and how these microbes can really drive like larger ecosystems, and that could be both natural systems, but it also could be managed like ag systems, correct? Yeah. So how would some of your findings, what are some of your findings that you think might be of interest to those in the ag community, whether ranchers or farmers?
Yeah. Yeah, some of the work we do, I won't talk about the water as much, but everything we're studying in water revolves around how does a stream remediate that pollution when it dries out and re-wet. So we're really interested in drought, how drought affects water quality. In the past, we've done some on drought and soil microbes, but just acknowledging drought is a very important, concerning topic that we're studying. I probably won't go into a lot of detail with you here. On the more applied side, everything we're working with in the soil system, even in the native prairie ecosystem, is very relevant to agricultural systems because at the microbial scale, a lot of the functions are the same. It's just the context is a little bit different. And came here to Kansas specifically to work in the prairie, which is also rangeland.
Yep.
The Flint Hills is just an amazing, again, coming from Wisconsin, where we have tiny, tiny, tiny patches of native prairie, or like one pasture in the corner of the property that was never improved or seeded. It's just spectacular to be here and see this huge extent of native prairie that was never tilled. And it's obviously rangeland and still in production. So everything we're doing in the prairie, I think, is relevant to rangeland. Specifically, we have a couple of projects thinking about how bison grazing and cattle grazing are similarly influencing the soil fertility and the diversity of the microbes and soils across the Flint Hills. And then we've also done work in the prairie systems, thinking about how fire and extra nitrogen change the soil microbiome. Of course, in a prairie system, you don't fertilize the prairie, but what the fire does is it makes nitrogen very relatively low in the soil, and yet prairies are really productive, which we know here very well. So we're interested in how adding external nitrogen inputs to that soil changes the microbiome function.
Could we go back to the bison and the cattle for a second? I'm just curious, are there differences in terms of how bison and cattle impact the soil microbiome?
There are similarities and differences, yes. So let's see, stepping back, when I talk to people about the bison versus cattle, I always remember that there were bison or buffalo, like I don't care if people say buffalo. It's American bison, also buffalo, across the whole North American continent in large numbers, only, a couple hundred years ago, and they were food for the people who lived here. So now we have cattle, which are food for the people who live here. There's a similarity, but not, they're not exactly the same sort of agricultural system, obviously. The animals themselves both have similar influence on the soil nitrogen and feedbacks to forage quality, in that as they eat, they digest the grasses, of course. And I have to throw in that, of course, it's also the microbes in their guts that are digesting the grasses. So plug for microbes. But without adding additional nitrogen inputs to the system, that extra digestion and then egestion out of the animal makes more bioavailable nitrogen for the microbes to decompose in the soil and go back up into the plants. And we see evidence of that sort of faster nitrogen cycling and higher nitrogen availability in soils under both bison and cattle. And that's a benefit to the system, in that it means more grasses and other, but forage grasses and all the other plants take up more nitrogen because it's available faster. The dung and other excreta from the animals, but especially the dung, it's more like slow release nitrogen, maintaining higher forage quality over longer periods of time, and having a sort of a positive feedback on forage quality. So that, I'm trying to think, we have more evidence just because we've done more work under bison for that feedback to be set up, where under bison, just on the Kanza Prairie right outside of town here, there is higher forage quality and more nitrogen in the soil and higher nitrogen availability. And then under cattle, we see higher nitrogen availability, less clear of a forage quality feedback. But that may relate more to the intensity of grazing than the simple, which animal do you have on the landscape? And our group hasn't done as much work thinking about other types of animals or mixed livestock situations, but qualitatively, like, in other words, this function is possible under any type of grazing system.
And then let's move to your other study you mentioned. I mean, I think generally when we think about fire, you view it as like a positive within a rangeland system. Could you talk a little bit about? Could you just break down a little bit more about the impact of fire on N and nitrogen within the system?
Yeah. Fire, clears out all of the residues, basically. The residues of the native grasses and forbs are fuel for the fire. They're full of nitrogen, carbon, other nutrients that would otherwise decompose back into the soil. So fire removes nutrients overall. The native prairie grasses and other plants are really good at fixing carbon dioxide from the atmosphere into carbon. They have huge root systems. They put a lot of carbon back into the system, more carbon relative to nitrogen. So over time, more carbon accumulates in the soil than nitrogen. It's, I guess, a characteristic of some of these native warm season grasses that they're more competitive with less nitrogen in the soil than some of the, a weedier grass or a crop production grass. So the advantage of having the grazer in the system, which would have, been the case for millennia with the bison, is that without adding external inputs of nitrogen, there's still faster nitrogen cycling in the soil and higher nitrogen availability to the plants. And that, overall maintains that higher forage quality without having to, go outside for inputs of nitrogen.
Yeah.
So both the fire and the grazing together help maintain that most productive sort of rangeland state.
Very interesting.
Yeah.
And then my next question was, do you have any ongoing studies where you're still looking at the results or future research questions that you anticipate investigating that may be of interest to those in the ag community? I think another way to say that is just what questions do you feel like in your field are still unanswered that might be of interest to those interested in regenerative agriculture?
Yeah, we and others have quite a bit of ongoing research. Where to start? I'll start by sort of comparing what I just explained about the animal influence on nitrogen to the fertilizer influence, and that'll kind of take us more into cropping systems.
Yeah.
So we have done studies in the prairie and in ag systems. You add ammonium nitrate or urea fertilizer to the soil, you might see similar effects of plants growing more, but the microbiome itself changes in a different way. So with the animals, I have had, for example, one student did an experiment where she sterilized soil and put dung back on the soil and showed that there are unique types of bacteria coming in through the dung. And that and some other factors make the actual types of microbes that live in the soil unique under grazing. When you add fertilizer, what happens to the microorganisms in the soil is, with excess fertilizer at least, and this is where the big question comes in, how much is too much? We don't know. But if you add a lot, that promotes the growth of microbes that grow really fast, but don't decompose the residues as well. So there's like this sort of opposite end of the spectrum of the type and level load of nitrogen added that can shift the soil microbial community into one that doesn't function as well inherently to like decompose the organic matter and release the nitrogen back within the soil. So too much nitrogen can shift the soil microbial community into one that is no longer doesn't have that inherent fertility anymore, for lack of a better turn of phrase. In ag systems, again, so we don't know where that level is. And that the level of too much, one reason we don't know or we'll never be able to recommend that this level is too much or this level is not enough, because every soil is different, which, practitioners all know, the soil type, what crop you put there, and how you manage it, really varies depending on the texture and the mineralogy and the history of that soil. there's a different microbiome in every soil too, just like, there's a different inherent everything about that different soil series. So for that reason, and also reasons related to historical management, we can't say in any particular system how much is too much. So what we're working on, myself and others, in individual case study locations, are field experiments that might help constrain sort of where the thresholds of losing the functionality of the microbial community are for that particular location or that particular climate type or soil series. That obviously requires a lot of work at each place, but that's not dissimilar from cropping trials in different locations, as your listeners probably know. And we're also working really hard on using our DNA-based methods to identify indicators of the healthier or more desirable functionality of the soil microbiome. So we do a lot of extract DNA from soil, sequence it, get a list of different types of organisms living there. Some of them we know might be the sort of weedier types. Some of them we know are the types that can decompose organic matter really well. Some of them are the types that associate with plants, like arbuscular fungi that have known really particular plant beneficial functions. Some of them are bacteria that can eat pathogenic fungi. And there's, a category, we can drill down deeper and deeper and assign sort of beneficial or detrimental functions to these lists of organisms that we sequence. And because there's so much complexity to these lists of diverse communities, we are still working through which specific indicators will be most useful to practitioners and producers in different situations.
I think that's a really big question we continue to get is when we talk about soil health and specifically soil microbes, is what are those indicators that we should be looking at? So I think that's really valuable work that you're doing. Is there any kind of initial things that you have seen, or I don't want you to get out ahead of the research, but I'm just curious if you have any initial thoughts of what are some good indicators for folks that want to better understand their soil microbiology and their soil microbiome, which of those indicators they should be looking at.
Yeah, I'll tell you what we're looking at and kind of how we're thinking about it. But with the caveat I already said, every soil is different, every field is different, every operation is different, and there's a lot of different factors that can influence the lists. I know folks can send their soils off to labs and pay for when I say a list, maybe some of your listeners have tried this or done this, and I'll say even myself, it's a lot of information that, especially without context, I would have trouble saying this is good or this is bad. As more of an expert in seeing a lot of types of data like that, I have the context kind of in my head to know what it's doing comparatively. But when we start a study like this, we try to identify some sort of reference point for that field or that that cropping trial in that area that we can use as a baseline of comparisons.
As in, you would compare it to a native?
Yeah. Just something that's less
disturbed. Yeah. Okay.
Some places that's easier to find than others.
Yeah.
But often, just a fence row or county line roadside that was never tilled, that can actually be pretty good. And you would know locally kind of what area is least managed, I'll just say. We're starting to look for different types of nitrifiers, which are really clearly defined. Those are the ones that turn ammonium, the bacteria and archaea, they turn ammonium into nitrate. In agricultural settings, this is not really, having a lot of nitrifiers is not desirable because they're competing with the plants for the ammonium. But they're everywhere, and there are certain types that are more greedy for nitrogen than others. So we can start looking at specific types of nitrifiers. We look for specific types of arbuscular fungi or pathogenic fungi or we call it saprotrophic fungi. That means those are the ones that are decomposing the residues. And we're definitely seeing in some of the trials we're working in some shifts in the fungi under different crop rotation series that are intriguing, but I can't tell you right now because we haven't actually gotten into the details of which type which type is shifting under soy after wheat after corn, but something like that does seem to have an influence on the soil diversity. And then the weedier types that are kind of more characteristic of excess nitrogen, I have a list of those as well. And I won't like list off the names because that would get especially boring. And but yeah, we have sort of categories of types that we have set up that we might think about. The relative abundance, like relative to a reference soil, or the relative abundance within one soil. So we have like a ratio or an index of something meaningful for nitrogen availability for plants or pathogenic susceptibility of that soil.
So you would say it's the balance of the microbes that is most important?
Yeah.
Yes.
Yeah.
Yes.
Not that it's the absolute number is important.
Yeah.
But as far as the methods and the data that any of us, myself, or anyone ordering a soil test gets back, those are all numbers relative to one another in the soil. They're not absolute numbers. So we have to think about it all in a relative sense to make it useful.
I think another question I thought of is you mentioned, with the grazers in the rangeland, that seemed to help with nutrient cycling. Do we have any evidence that we would expect a similar relationship with grazing and integrated crop livestock systems?
There's a few studies on that. I have one small case study ongoing on that. And of course, integrating livestock back into the fields, or even just applying manure back into the fields. We do this fairly often in Wisconsin, but the larger the operation, maybe the more complicated it gets. Sort of theoretically, based on everything I explained earlier, I see this as a sort of a viable or very interesting, management strategy to maintain, I don't want to say fertility, but, nitrogen availability without extra inputs. And also, I would predict that we have less of a chance of flipping that microbiome into that weedier state that doesn't decompose residues as well. When you add livestock or add manure, you're not just adding more bioavailable nitrogen, but the organic matter along with it. So any sort of organic amendment is more likely to maintain that functionality of the microbial community. So there are some studies that show evidence for that, but there are just fewer studies overall. Yes, I'm very interested in that myself. I think it shows promise.
Yeah.
And we definitely are, I'd say, on the indicator side of things, we might be able to pull out indicators of the microbiome changing with animal inputs in the system, So, a relevant question might be for how long or, how frequently, how much, how often do you need to put livestock in on the field or whatever in order to get some beneficial function back into the soil. If we have a microbial indicator of that influence, we could track sort of how long that influence lasts. The microbiome changes a lot more slowly than even I sort of first anticipated coming into a lot of this research. It's less sensitive, but also more robust or more sort of sustainable once you get to a certain point.
So it's harder to make that change, but once you make it, in theory, it's a little bit more in place. It's harder to change it back, I guess. Is that?
Both sides of sort of
this. Very interesting.
Yeah.
Well, Lydia, thank you so much for coming on. And the final question I'd just like to ask all my guests is kind of to turn it back over to you and let you just talk about what's exciting you the most in your work currently, whether that's in your broader field, whether it's with research or the classroom. I just really want to open it up to you so people can know what excites you most in your work currently.
Yeah, there could be so many answers to that. I'll stick kind of on topic and just tell you about diversity writ large, because this is a huge question on the basic biology side of this topic. How much diversity do we need in a microbiome to make it functional or useful? So, we have exciting work in this area, and others do too. But some of our more recent work has shown we actually took soil, sterilized it, put microbes back into it, and diluted that diversity of that microbiome down like orders of magnitude from 10,000 different types of organisms down to 1,000 or hundreds. And showed with the experiment that there is loss of function when you get down into these really low levels of diversity. But up at the more normal levels, the absolute number of diversity did not predict the functionality. And I don't know, this is exciting. The graphs I'm imagining in my mind, I can't show you, but it's evidence that diversity does matter. And why that matters is the more different types of an organism that can provide nitrogen to your plant, the more resilient that whole community is to shifts like under drought,
precipitation,
tillage, or any sort of disturbance that you must do when you're managing a crop field. So diversity is good, but there's nothing. But there's a huge range of normal when it comes to diversity in which most or many crop fields do fall. It's not. There's nothing about an agricultural system per se that has to be sort of depauperate of microbial diversity or function. And that's when we get back to everything we just talked about, like which specific types are most important. But I say that just because it's kind of an exciting, motivating, question on the basic side of our research, not the applied side. But also kind of as a reality check that we'll never have. Microbial ecologists or microbiome folks, there might be, magic dust out there, here, there, and everywhere. But there will not ever be like one number or one gold standard of diversity, but more diversity is better. We can say both things with confidence. So that's, I don't know, that's what motivates us on the basic side of things too, I guess. And I would also put in a plug for basic biology because I already mentioned, like when I went to college, I avoided the heavy duty biochemistry classes and organic chemistry classes and things like that. But again, once I start thinking about how does decomposition really work and how do those residues turn back into quote unquote fertilizer, I needed to learn a lot of that stuff. I went to grad school to learn a lot of that stuff. Not that people should go to grad school, but just acknowledging that there's so much more to learn and the basics. Even if you don't love biology in high school, some of those basics really do matter. It's never too late to pick any of it up. And I tell students this too, even if they don't go into the nitty-gritty details of everything I was sort of scratching the surface with in the past half an hour. Just appreciating the basics, I think, can go a long way. So, but and get out there and get your hands dirty,
Well, I just would say that in my experience with going out and interacting with farmers, particularly the farmers that have been thinking about these questions for longer and trying to do things to improve their soil health and do regenerative things, it feels like the thing that you hear them talk more and more about is the biology. I think there's a recognition of like, if you don't, like, there's something very fundamental about having to realizing this is a biological system and being able to have at least a baseline understanding of how you're interacting with your biology when you're making changes in your system. So I would just say you're not alone in that. I think there's a growing trend within the agricultural community to I even had a farmer just straight up tell me, is like, if I could have gone back, I would have taken more microbiology classes. So I think there is a growing momentum in that area. So we really appreciate you taking the time to come on and explain some of those things to us. Hopefully, maybe spark some questions for our audience and maybe some things that they can look at. And maybe as you get some more of those results coming in going forward, we can have you back and hear more about what you guys are doing. So.
Yeah, absolutely. Yeah. I'm sure people can find me online if they have other email me if they have other questions. I'm not sure how commonly that happens. But yeah, I'm right here and could go into more detail if it's of interest. I'm happy to. Um, yeah, thanks again for having me, and thanks for doing the podcast series.
Yeah. Well, everybody, thank you for tuning into the K-State Regen Ag Podcast, and we'll talk to you next time.
Oh, give me a home where the buffalo roam, where the deer and the antelope play, where seldom is heard a discouraging word, and the skies are not cloudy all day. Home, home on the range, where the deer and the antelope play, where seldom is heard a discouraging word, and the skies are not cloudy all day.